Today I'm here to talk about bioengineering of animal species. Now, what is bioengineering of animal species? Well, I'm here to explain that today. So, you may have heard of it as transgenic or genetically altered or genetically modified animals. A genetically engineered animal can be defined as an animal that has a change in its genes or DNA. This change can occur by addition, deletion, substitution of an animal's genetic material or makeup. This process is done by transgenesis, in which new material is genetically transferred from one animal to another. However, we no longer have to use the transgenesis method due to new advances, and instead we just delete and manipulate their present, whether it's past or current genes. For an animal to be considered genetically engineered, they must experience induced mutations, for an example, like chemicals or radiation, in which it has to be direct. There happens to be 3 different types of cloning techniques. There happens to be therapeutic cloning, reproductive cloning, and there happens to be DNA cloning. If somebody intends to mutate an animal's genetic DNA to another existing animal's cell, whether it was previous or current, this is called reproductive cloning. An experiment took place where we took the genes of a sea anemone, a zebrafish, and a jellyfish, where we found to create fluorescent proteins. This is how the genetically engineered sea animal, glowfish, came to be. It is a fish you could buy as a pet that was marketed in the early 2000s. It was soon enough that the household companions were seen as immoral or ethically wrong. It soon enough sparked a controversial debate and were seen with a lot of ethical issues. It became so serious to the point that the state of California decided to ban them. The reason being is it sparked a lot of ethical issues. The first one happens to be large numbers of animals used. The second is invasive processes, and lastly, it must create some sort of limit within the biological engineering aspect. There is a variety of agricultural techniques which can range on the spectrum for things like resistance to disease, environmental sustainability, and food quality. Productivity of farm animals can be increased using genetic engineering. Such as pigs and sheep, they are genetically changed to particularly develop higher levels of growth hormone. We have found that we can genetically engineer farm animals for beneficial reasons like increasing the quality of food produced. Some people may choose to genetically engineer an animal such as a pig due to increase higher levels of omega-3 or to use organs. That is why when the nutritional value of a food product goes up, don't be surprised when the price goes up as well. Genetic engineering has been used to reduce the rates of agricultural pollution. The best example of this would be the EnviroPig. It is a pig that was genetically engineered to produce an enzyme that breaks down phosphorus within the metabolic process. It then limits the amount of phosphorus that is released in the manure afterwards. Animals are highly used in medical institutions to help humans with testing with antibiotics and advanced medicine. Lots of hard work goes into this kind of research every day. Genetic engineering has opened many doors to the creation of many human disease models that were unavailable before. Although I did put that these processes are legal in Canada, these are prohibited in Canada. We are allowed to do research to help with experimenting, which brings me to my next topic, xenotransplantation. Xenotransplantation is the process in which we transplant cells, tissue, organs from animal donors to people. However, this topic tends to be very controversial due to the fact that there could be a potential disease transfer. That's everything for today. Thank you for listening.